The answer project is a very important topic. Actually, it's a very hot topic worldwide because the antibiotic-resistant bacteria is spreading and it's increasing the number of deaths worldwide. And this project it deals with different ways to treat wastewater to remove antibiotics and antibiotic-resistant bacteria. And also we are studying the spread of these antibiotic and antibiotic-resistant bacteria during the water reuse. In fact, water reuse it's very important because a lot of countries worldwide they suffer from water shortage, and water reuse is a very important source of water and can be used for agriculture and other practices. In the ANSWER project, we are studying the uptake of antibiotics and antibiotic-resistant bacteria by crops, and also the risk associated with the consumption of these crops irrigated with reclaimed water. In this project, there are 15 early stage researchers, and in the CSIC we have two PhD students, Đorđe coming from Serbia, and the other one is coming from Portugal, Francisco Diego de Almeida.
My name is Đorđe. I am an ESR number 8 within the ANSWER ITN project. Topic of my research is uptake of antibiotics and other antimicrobial compounds by crops, specifically vegetables, the one that we will eat them raw. Among the 15 ESRs that are the part of the ANSWER project, I'm dealing mostly with the chemistry and with the health risk assessment. Marija Sklodowska-Curie Innovative Training Networks are mainly focused on young researchers, early stage researchers, and in our project there are 15 of us. We come from 10 different countries, and at the very beginning of the project we were a very professionally diverse group, and now I could say that we become a multitasking team. We consist of engineers, chemists, biologists. We deal with many different subjects, and this allows us to have one comprehensive overview over the issue that we are dealing with it, and it is reuse of wastewater for irrigation and uptake of antibiotics, transformation products, metabolites, and assessing the health risk for the environment and for the humans. 15 of us, we are also PhD candidates. We all have our subjects that we are dealing with, which are very innovative, and they should provide a solution on the water scarcity issue and the reuse of wastewater for irrigation. Nowadays we have to use wastewater for the irrigation. It's not a matter of choice anymore. And the reason is water scarcity. Water is a very limited factor, and the demands for agriculture become higher and higher. So we have to. And the current technologies, they fail to remove antibiotics and other pharmaceuticals. So when we use treated wastewater for irrigation, we also have antibiotics in that water, which eventually will end up in the soil and then will be uptaken by the roots and translocated within the plant and end up in our plate. For my project, we did several experiments, and also we did the monitoring studies of vegetables here from Spain, from suburban area of Barcelona. What we did is that we collected vegetables that are commercially available, that are eaten raw, and that they were irrigated with treated wastewater. So we analyzed lettuce, tomato, cauliflower, broad beans, different types of vegetables, and also the soil. And the results shown that we could detect and that antibiotics are uptaken by those kind of vegetables. And also they end up in the soil. And actually we had the difficulties to find a control zone, the one that was not polluted with the antibiotics. As I said, pharmaceuticals will remain after the treatment. When we apply this water to the soil, antibiotics, some of them depends on their physicochemical properties, will be uptaken by the roots and then translocated, so some of them, not all of them, and then they will go from the water, soil, roots up to the vegetables. Now we are investigating which from all the antibiotics that we use, not from all but from majority that are widely used, which of them they can be uptaken and translocated. So we are interested mostly in those ones that they will end up in the fruits that we are eating. Until now we found out that they will be uptaken and translocated highly by lettuce, also by tomato and by the cauliflower. So within our project we had to disseminate our research, our results to the public, to the society, which I found very valuable because data visualization become very important, especially nowadays when the information can be spread easily. So we did several dissemination activities. We visited high schools, we gave a talk, tried to educate the kids to tell them about the water scarcity issue. Also we visit end users, farmers. With them we spoke about the reuse of wastewater, what could be the challenges, the risk, what could be the alternatives. Also we try to use media to disseminate our results and to inform the public about this issue. So I gave a talk in Slovakia, in Bratislava, in their radio. Also we wrote some articles for the popular science magazines. And this is one of the ways that we would like to disseminate our activities and to share what we found out with the society in order to raise awareness about this hot issue. Marie Curie ITN is a very, let's say, life-exchanging experience, professionally and personally as well. And within this project we traveled a lot, we visited other labs, we went to several trainings and conferences. And in my case I spent 3 months at the University of Cyprus, where I did my secondment. There we perform unique experiments, real field experiments, where we irrigate cucumbers with treated wastewater with different kind of treatment. Also we fortify this irrigation water with antibiotics. We apply biochar in order to reduce the uptakes, and everything was done in the field scale. It is all this travelings and spending time in another lab, in another place, in another country. It's very valuable and also delightful. Within this, in this way you have opportunity to share the knowledge, opinions, learn some tricks, make friends of course, live in another town, country, culture, have an interaction with the people that are not from your comfort zone, and it was very nice experience. Another secondment that I did was in Bratislava in Hikem. It is a private company, and this was also a very unique opportunity to see how the private sector is cooperating with academia closely and successfully, to have an insight how the private sector works, to also to do some remarkable experiments with the state-of-the-art instruments, and to learn from them how to process the data, how to elucidate the structure of unknown compounds, which they're experts in, and also it was very nice and remarkable experience.
So hello, my name is Francisco Cerqueira, and I'm an early stage researcher at the Institute of Environmental Assessment and Water Research in Barcelona, and I'm doing my PhD studies here. Mostly I work with bacterial populations, and I also work with antibiotic resistance genes in crops. So the ANSWER project is of extremely importance, since nowadays we have a problem of water shortage, and treated wastewater is being used more and more. So we need to study the impacts of its utilization, and in irrigation, for example, that it's utilized in farms, and to check if it's contributing to the spread or not of antibiotic resistance. So antibiotic resistance is when bacteria change in the presence of antibiotics, which are medicines that are used to kill them. And this can be intrinsic or acquired. Acquired can be by transduction, transformation, or conjugation. Conjugation, let's say, it's when bacteria exchange genetic material with the cell-to-cell contact. So they exchange material, they touch each other. And transduction is when the genetic material is exchanged through a virus. And the transformation is when bacteria are able to uptake genetic material from the environment itself. So conjugation is when bacteria are able to exchange genetic material between themselves through direct contact. It means that they touch each other with a structure named pilus, and then they can exchange the genetic material. Transformation is when bacteria in the environment are able to uptake DNA that's present there. Antibiotic resistance happens when bacteria are in the presence of antibiotics. So, and this can happen everywhere and even occurs naturally, since antibiotics are a way of the organisms to outcompete each other in the environment. And it also happens a lot in hospitals, in the community, or even in the farms. So in the farms, for example, animals are treated with antibiotics and they may get antibiotic resistance bacteria. In the community, people are treated with antibiotics, and then again they can transmit antibiotic resistance bacteria to each other. And in the hospitals, due to the utilization of high dosages of antibiotics, so it's a selective pressure, and only bacteria that have genes that give them resistance to antibiotics are able to spread there. So, but not all bacteria are, let's say, bad, and actually they are necessary for plant growth. And there are lots of bacteria that are named plant growth promoting bacteria, which, for example, they can fix nitrogen for the plants, or they can even help to boost the plant defenses against other bacteria, pathogenic bacteria. So molecular methods are used to study DNA, RNA and proteins, for example. And in my case, since I want to study specific genes, some antibiotic resistance genes, I use a method called quantitative polymerase chain reaction, or mostly known as qPCR, and that allows me to check the presence of those genes in my samples and also quantify them. There are also other methods like DNA sequencing that allows to know the exact order of the nucleotides present in a genome, and with that you can know the genes, know which bacteria are present in a population. So in our project we have to perform outreach activities. That means that we need to show people what we do and why it's important to invest in scientific research. It was very good for us because it made us to develop some communication skills, since we have to tell non-scientists what we do. And for example, we visited schools, from elementary schools to high schools, and the way you have to tell things is completely different. So in our project we attended several secondments. It means that we have to spend some time abroad in several other institutes, and it was great for us since we made new collaborations, worked in different environments, check how people worked in different countries, and made us also to plan for our research during a few months.
When we went to do one of our dissemination activity in a high school here in Spain, where we held a masterclass for many students, I realized that the crucial part, what is missing, it's education. So these things should be incorporated in schools, and we should educate kids, and also we should find a way to listen educated people. And that is one of my biggest impressions from all these dissemination activities that we did.
So I have to say that I agree with Đorđe said, that education is very important, but more important than that, I think it's to spark the interest of kids in science and scientific research. Because there is a myth that science is too hard, maybe sometimes it's not worth the effort, and we need to take that thing out of the people's minds. I think it's hard, but it's doable and it's very interesting. So you have to create interest in at least young people to appreciate science.
The ANSWER project is a network framework. That is, the main idea is to treat the problematic of antibiotic-resistant in agricultural uses, especially connected with water reuse. This is a much broader subject, but the specific way the ANSWER targets to this problem is making a very broad network of different labs with different approaches and different techniques, and with a very good and selected team of ESR, early scientific researchers. And this is a very new system, and at least for me was a new experience, because I'm not really connected with obtaining a degree like I'm more used to, but is focused to getting abilities and connections. And this is a very nice approach, and I think it's very important to keep on doing this kind of networks.